Over the last few decades, there has been rapid growth in the field of molecular biology and biotechnology. This growth is a result of intensive research undertaken in these fields with the help of transgenic animals. The genome of transgenic animals is deliberately modified to possess and express an extra foreign gene. Over 95% of transgenic animals are mice. Before the development of molecular genetics, the regulation and function of mammalian genes could only be studied through the observation of inherited characteristics or spontaneous mutations. The science of genetics developed quite late. However, breeding animals for purposes such as increased milk production was known to man for a long time. The first genetically engineered animals were the chimeric mice produced during the 1970s. They were created through an experiment in which cells of two different embryos of different strains were combined together at an early stage of development. Therefore, a single embryo was formed and implanted into a surrogate mother who gave birth to chimeric mice. These mice exhibited characteristics of each strain and became the first transgenic animals. Let us now understand the main reasons behind the use of transgenic animals. Some transgenic animals can be especially designed to study gene regulation and the effects of genes on the normal functions of the human body and its development. For example, they can help in the study of the biological role of the insulin-like growth factor. To study the role of insulin in humans, genes from a rabbit or a mouse are introduced into another mouse, which then gives birth to transgenic animals having the altered gene for insulin. Then, the biological effects of this introduced gene are studied. Thereafter, information is obtained about the role of insulin in the human body. Transgenic animals are also designed to understand how genes contribute to the development of a disease. They serve as models and help in the research of new treatments for diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. Transgenic animals are used to produce expensive biological products such as alpha-1 antitrypsin at a cheaper rate. Alpha-1 antitrypsin is required for making medicines for the treatment of human diseases such as emphysema. Attempts are also being made to create biological products for the treatment of genetic disorders such as phenylketonuria or PKU and cystic fibrosis. In 1997, the first transgenic cow, Rosie, was created, which produced human protein-enriched milk containing 2.4 grams of human protein in every liter. This milk contained the human gene alpha-lactalbumin, which made it a more nutritionally balanced product than natural cow milk. Therefore, the milk of the transgenic cow Rosie could be given to babies and elderly people with special nutritional and digestive needs. Nowadays, transgenic mice are being developed to test the safety of vaccines such as the ones meant for polio before they can be used on humans. In future, if testing on mice proves to be successful and reliable, it could replace the use of monkeys for vaccine testing. Transgenic animals are helpful in testing the toxicity of chemicals as the results can be obtained in a short time. For this testing, specific transgenic animals are designed which carry genes which make them more sensitive to toxic substances as compared to non-transgenic animals. Thereafter, they are exposed to the toxic substances and the effects on them are recorded and studied. The creation of transgenic animals has reduced the overall use of laboratory animals. For example, there is a shift from the use of higher order animals such as dogs to the lower order species such as mice. However, use of transgenic animals is still surrounded by a lot of ethical issues.